Good afternoon. We are going to try to finish lipids and proteins today. I think that's all the time we have. I did not get to nucleic acid with my other class, so I don't think we will get there either. But that's okay. Your quiz on Thursday is only up to and including lipids, which is the first thing we are going to do right now. Now, I would want to start with giving you a summary first for lipids before we go into the PowerPoint slides. Okay, so if you can take out a piece of paper, you can put the word lipids in the middle, and then I'm gonna draw different examples for you. Okay, here we go. Lipids. Now, the four types of macromolecules include carbohydrates, proteins, nucleic acid, and lipids. And I want to remind you that lipids are a little bit different compared to the other kinds of macromolecules. Specifically, all the other kinds of macromolecules are essentially polymers, very long chain of repeating units, okay, that we connect together like a train. However, for lipids, that is not the case. Okay? Lipids are not polymers, but rather they are made up of several components connected together. Okay? So I'm going to start with the first lipids here. They are not in no particular order. Okay, so the first one we're going to talk about is triglyceride. Triglyceride. Okay, please learn how to spell that properly. Triglyceride is just the fancy word for fat. If I do ask you about it on the quiz, please write triglyceride, okay, instead of fat, because that's the proper term for it. And in order to build a fat molecule, you would need to have two things, right? Please use some colors as well. We have this component called glycerol, and we need to have three of these zigzag stuff. So these things are called fatty acids. Okay, please write these down and leave it next to you when we go to the PowerPoint slides, okay? It will help you understand it a little bit better. Okay, so like I said, it's not a long chain of repeating units like we saw with cellulose, for example. Instead, we need two components. We need one of these things called glycerol, and we need three of these tails called fatty acids. And that makes up a fat molecule. That makes up a triglyceride. What do we need fat for? Basically, fats are useful for long-term energy storage. Okay? And they are good for insulation and protection of organs. Okay? Insulation means it keeps the organs warm, particularly like a day to day, right? It's like minus whatever, like 25 or something crazy, right? And we want to keep our internal organs warm. And fat is excellent for doing that. Fat also serves as a little bit of bubble wrap that goes around the organs. So, if you bump yourself, you fall down, your kidneys, your hearts, they are going to be nicely cushioned. So that's triglyceride. Okay. For the purpose of the quiz, please recognize what a triglyceride look like. There's a question like that in the study guide. Remember the components you need to build them, as well as the functions of these things. All right, we have a second kind of lipid. It is called phospholipid. Phospholipid. How do we build a phospholipid? We need this guy again. This is a glycerol. And this time, instead of having 3 of these zigzag, we only need 2 of them. So 2 fatty acids. And we need a new thing here. Okay, this is called a phosphate group. Okay, this is just a summary for you, okay? I'll explain in more details when we switch to the PowerPoint slides. Okay, so this thing is called phospholipid. And the function is that it is the major component of the cell membrane. The cell membrane is this thing that goes around your cell. It separates the living from the non-living. And we will learn more about it later on in the course. But it's made up of phospholipid. Yes.
So for the triglyceride, it's glycerol plus 3 fatty acids.
Yes.
This one is the glycerol plus phosphate.
And
The
2
two
fatty
fatty acids.
Okay, sweet, guys. Thank you. Okay, next. We have wax. Okay. Wax is like this, okay? Okay, so by now you know what this is, right? What is this thing? Yeah, fatty acid, right? Yeah. So wax is like a long chain of fatty acid. They could be, short or long, doesn't really matter. And this part is weird, okay? Like you just have to believe me, okay? Like you will learn this in chemistry later. I know I keep on saying that, but you will. There are these OHs that are attached to it. And whenever you have OH attached onto a molecule, we actually call them alcohols. Okay, so it's fatty acid with a bit of alcohol. Not all alcohols are like drinking alcohol. Drinking alcohol is ethanol, right? But you have other alcohols too. Okay, your windshield fluid is actually a kind of alcohol. You can't drink that. There's a little skeleton that tells you it's poisonous, right? You have like rubbing alcohol, and what makes it an alcohol is the presence of these OH in the molecule. Okay. I wouldn't worry too much about wax for the quiz. Okay, I'm just putting it here because we'll learn about it, but don't worry too much about it. What do wax do? They form water repelling surfaces. Okay, like the skin of an apple, like your skin. When you wash your hands, the water don't like go right through the skin, right? At least it shouldn't. It's because of the wax. Okay. Just one more here. Steroid. When you hear the word steroid, maybe what comes to your mind is doping, right? Drug that would enhance performance for athletes, for example. But your body actually makes steroids, and we need steroids for normal bodily functions. And steroid goes like this. Okay, you can tell you are looking at a steroid if you see these carbon rings. They're kind of like honeycombs, if you would. Okay, so there are always 4 carbon rings. And then you have other stuff that are attached to it, okay? Depending on what kind of steroid you're looking at, you will have these extra things that are attached to it, and they are called functional groups. Okay, so I already introduced to you, if you attach an OH, then it makes it an alcohol. It turns out if you attach, I realize it doesn't look too clear here. This is like a oxygen right here. Then it makes it into something called ketone. Okay, it doesn't really matter. Okay? The key thing here to know is depending on what kind of functional group you attach to the carbon ring, it is going to give it unique property. Okay? The carbon rings is like your phone, okay? You buy your phone and then you buy a bunch of accessories, right? Where you buy the screen protector, the case, the little ring that you put on the back so that your phone can stand out, right? All those things are accessories to your phone. So these are accessories to the steroid, and it's going to give it unique properties. You guys understand? Okay, so what do we use steroids for? Steroids are also part of cell membrane. And they form hormones. Okay, hormones are chemical messengers in the body. It allows different parts to communicate with each other. Example of steroid hormones would be sex hormone. Does anybody know examples of sex hormone, either in male or female? Just yell them out. Testosterone, that's the male one. What about female? Yeah, there you go.
It's progesterone,
Progesterone is another one. That's right. Very good. Okay.
but that's in the same.
Yeah, you can put
Okay.
you can put progesterone here as well. Okay.
Okay.
There is also aldosterone and a bunch of them. Okay. Let's get put through it. Okay. So for the quiz, please be able to identify steroid, fats, and phospholipids. Don't worry about wax. I'm not gonna quiz you on wax. Okay. You gotta know the components that makes it up, and you gotta know what it does. Okay? If I ask you to list one function, then please just give me one function. All right. Like I know you know all of it, but just give me one. Because sometimes you put too many, and then it turns out one of them is wrong. Then I'm forced to take marks off, right? Okay. Just you put one if I ask for one. Got it?
Oh,
You sure? Go ahead. Yeah.
for how long is the
40 minutes. 40 minutes.
But like how many questions?
Okay. Since you asked, let me just reiterate this to the whole class. If you go to the quiz folder, there's the quiz information, okay? Click on that. 15 multiple choice, 5 short answers. Okay? Yes.
I was checking, but do we have to draw the diagram, or are we gonna give it something like 95 or 2?
Oh, someone opened the door again. No, I'm not gonna ask you to draw anything, okay? The only thing that you should know how to draw is the thing I showed you last class with the molecules joining together. Yeah. Huh? The quiz includes the lipids. Yes. I'm sorry, but it does. Yeah.
We were talking about the monomers.
Huh? Yeah. Like this stuff, right? Remember? Yeah. Remember we drew this last time? Right.
Oh, yeah.
Yeah. Okay. If you don't know what I'm talking about, guys, go watch the review video. Okay. Seriously. Okay. And do the self quiz. Okay. I don't know why you guys are not doing it. Look, there's a self quiz. Okay. How many of you have done it? Let's see. 14 of you have done it. We have 35 people in the class. Okay. That means 23 of you have not done it. Okay. Please do it for your sake. Okay. For quiz 1, lecture 1, and lecture 2 as well. And then also the review video. Okay. Review video. Only 13 of you have watched it. The quiz is on Thursday. Please watch it. Okay. See, if you don't do well on the quiz and then you come to me after, oh, please tell me what I can do better. Watch the video. I'll see if you have watched it, right? Okay. Seriously, the video will help you. Who watched it? Put up your hand and watch it. Okay, tell the rest of the class, was it useful? Yeah. You're not just making me feel good, right? It's actually useful because everything in the video will show up on your quiz. Okay. I am very transparent when it comes to what's on the quiz. Right? Because it's biology. You're gonna have to memorize it. Okay. Even if I give you the exact same paper, as I mentioned in the video, you still have to go through the effort to memorize the stuff. Right? Okay. So that's why. Please do it. All right. Back to the PowerPoints. All right, lipids. What are the key elements of the lipids? Well, we have the CHO again. Remember, we also have CHO in the carbohydrates? Right? Well, here they are again. And in addition to the carbon, hydrogen, and oxygen, there are also trace of phosphorus. A little bit. Okay? Not a lot. I already mentioned to you, unlike other macromolecules, lipids are built with components rather than repeating units over and over again. Okay? All lipids are hydrophobic. What does that mean? That's right. They're fear with water. They don't mix well with water, right? Okay. Carbohydrates are hydrophilic, but lipids are hydrophobic. You know that already because when I asked you for examples of hydrophobic molecules, you told me things like oil, right? Wax. Let's start with triglyceride. Okay. Triglyceride, as I told you, have the summary sheet next to you. You will see we have one glycerol, and the molecule looks like this. Okay? I'm not going to actually draw it. Don't worry. The key thing to know is there are 3 OHs here. And I'm going to connect it with 3 fatty acids. Right? So what's the name of the chemical process we use to connect things together? If I have 2 monomers and I want to connect them together, what's the name of the process? Exactly, right on. Right? Dehydration synthesis, right? Where you'll be removing a water molecule. So here I will remove 1 water molecule, second one and a third one, and then you form the bond, then you have the triglyceride. Okay, the answer is actually right here. I didn't even know. Okay, dehydration synthesis. So now we create 3 water molecules. If you want to break it apart, the opposite of dehydration synthesis is hydrolysis. Okay. So 1 glycerol and 3 fatty acids. The fatty acid comes in 2 variations, depending on whether it has this equal sign looking thing in it. Okay. If you learned chemistry before, you might know that this is called a double bond. If not, you will learn later in the program. So if the carbon does not have the double bond, if the carbon does not have the equal sign, then it is going to have a maximum number of hydrogen connected to it. On the other hand, if it has the double bond, then it still has room for more hydrogen. Okay? So no double bond, we say unsaturated. And if it has no double bond, then we say saturated. Let me show you. Oh, sorry, before that. Where do we get our food, get our fats from? I mean, it's kind of everywhere, right? Like most of the food we eat would have some kind of fats. People don't deliberately, try to consume fats for the diet. It's just kind of part of your cooking, right? So butter and oil obviously are high in fat content. There are some good fats as well. Fish has good fats in them. Walnuts and all these things have healthy fats. Avocado, right, for example. All right, so let's take a look at saturated fats. Try not to confuse between fats and fatty acids. Okay, a fat is the whole thing. That's a fat molecule. Yeah, the fat molecule contains fatty acids. You understand the difference? Yeah. Whole thing is fat, and the individual tails are fatty acid. All right. So what makes it a saturated fat? A fat molecule is saturated if all three of its fatty acids are saturated. Okay? So if you take a look at this one, there are no double bond, right? There are no equal sign in it. And the tails are saturated, meaning they have the maximum number of hydrogen atoms. There are no more rooms to fit more hydrogen on them. That's what they're saturated with. Okay? You cannot put more hydrogens on them. And they tend to be solid at room temperature. And here is why. Okay? You have, this is the glycerol, and then you have the tails, which are pretty much straight. Okay? Straight. And so I can put another fat molecule next to it, and they would be able to get really close to each other. Okay? So they are able to pack closely with each other, and therefore they tend to be solid at room temperature. It's like if I ask you guys to line up against the wall here to fit everybody, you kind of have to put your hands down, right? Okay? You don't want to be sticking your hand out and preventing people from getting too close. You understand that? If it packs really closely at room temperature, it turns out they also pack kind of closely inside your blood vessel. Okay? So the saturated fat is not something you want to consume too much of. Okay? Most animal-derived fats are saturated. So we're talking about butter, lard, that's like the white stuff people use to make pastry, right? Bacon grease, all these things are saturated. And you want to minimize consuming those things. Okay. Next, we have unsaturated fats. Remember, fats is the whole thing. What makes it an unsaturated fat is if at least one of the three tails are unsaturated fatty acid, then the whole thing is an unsaturated fat. How do you tell if the fatty acids are unsaturated? What are you looking for? The double bond, right? You will see the equal sign, right? More importantly, whenever there is the double bond, it causes the tail to have a sharp bend, okay? They call it a kink in the tail, right? And so, if you draw an unsaturated fatty acid, you have the glycerol here, let's say two of them are saturated and the third one has a kink, then a second molecule is not going to be able to get very close to it, right? It's like lining up here, but sticking your feet out or sticking your hand out. The other person is not going to be able to get very close to you, right? And if the molecules can't get close to each other, then they tend to remain as liquid. Right? At room temperature, yes, and also they don't tend to pack as easily inside your body, and therefore it's relatively healthier. Okay, most plant-derived fats are unsaturated. So think about your cooking oil, right? What do you guys use to cook? What kind of oil? Olive oil. I heard something else. Canola oil, right? Avocado oil, grapeseed oil. All these things are unsaturated. Okay. Now remember at the beginning of the course, I told you we try to fit things into one category and then another, but sometimes there are exceptions, right? Yeah. So usually this is where some people would say, oh, I cook with coconut oil, right? And coconut oil actually is not liquid, even though it's a plant derived fat. Okay? Coconut oil turns out to be very high in saturated fat content, and so it's more like a solid or like a paste type of state. Okay. So don't use things that are obscure like that on the test or quiz. Right? If I want you to give me an example of this, just use the ones I told you. Okay. Olive oil. That's easy. Got it? Now, there are some essential fatty acids that we need. And essential means your body cannot make it, but you need it for normal body function. Examples of essential fatty acids, I'm sure you have heard of them. Omega-3, omega-6 and 9, right? So there are different examples of these things. You don't have to memorize it. Omega-3, the most two common ones are EPA and DHA. You might have taken those as supplements, if you have kids, right? During pregnancy, it's usually recommended to take EPA and DHA because they are beneficial for the brain development of the baby, okay? Also, the omega-3, 6, and 9, they have shown to be good for cardiovascular function. And so even if you're not pregnant, sometimes you might be asked to take these things. But like I said, generally you shouldn't start taking supplements on your own. You should always consult your doctor and see if you actually need it. Okay. A lot of times you already have enough of it, and taking more, you're just gonna end up excreting it from your body, and it's a waste of money, right? This is just to show you what they look like, right? They are very long tails, right? With a lot of the double bonds. And that's what EPA stands for in here. You don't have to memorize it, okay? I'm just showing you. Okay, one final picture to show you the differences between saturated and unsaturated. What is this one? Okay, is this saturated or unsaturated? Now, how do you know what gives it away? The kings, that's right? The bench, right? Okay. In fact, this one has 3 unsaturated fatty acid tails, right? You only need 1 to make it an unsaturated fat, right? But you can have 2 or 3. The more unsaturated tails you have, the healthier it's going to. Okay, something like this is called polyunsaturated. Okay, and you can see this guy, it's like pretty much straight line, right? But they both have the, what's this component called? Yeah, the glycerol, right? Glycerol and 3 fatty acids. Are we together? All right. Trans fat, okay? Trans fat is the worst fat of all fats, okay? How do you get trans fat? Trans fat is a byproduct of a process called hydrogenation, okay? So the idea goes like this. Right? I already told you unsaturated fat is like healthier, right? Yeah. But it doesn't make sense to pour oil onto your toast in the morning. Right? It just doesn't, make sense or feel right, right? And so they've invented a process called hydrogenation, where you take the unsaturated fat and then you add more hydrogen to it, and then it becomes semi-solid, like margarine, right? That you can spread, or shortening that you can use for baking, right? And in that process of converting the fat, you create trans fat as a byproduct. Okay, so in case you missed it, you start with unsaturated fat, you add hydrogen to it, and then it becomes saturated, right? In the process, we create trans fat. Okay. Trans fat are commonly used in many fast food or processed food because number 1, they make the food taste better, and number 2, it increases the shelf life of these products. You can keep them longer without spoiling them. However, your body cannot break down the trans fat. Trans fat goes into your body, it doesn't come out. It stays in your body for a very long time, and it's linked to increased risk of heart diseases, diabetes, obesity, high blood pressure, and it's just really bad for you. Okay? Yeah.
Is it wrong from the usage?
Absolutely. That's my next slide. Okay. So because it's so bad, the Canadian government decided to ban it in 2018. Okay. So no worries. You can eat more junk food now without worrying about trans fat. Of course, they're still bad for other reasons, but it's at least you don't have to worry about that. Your question, Casey?
Is butter trans fat?
Butter is not trans fat. Butter is a saturated fat. Okay. Margarine, because of the hydrogenation, it would contain some trans fat. Are we okay? All right, I hope so. Almost there. Number 2, right? If you have your summary sheet, right, you can follow along. The second kind of lipids, it is called phospholipids. Okay, now phospholipid is a very important molecule that we will come back again in the next couple of lectures. But for now, I know it's a little bit hard to tell, but there is the glycerol right here in the middle. And then you have the tails, right? And you have how many fatty acid tails do we have this time? 2 as opposed to 3, right? Okay. If we focus on the left one, do you think that's saturated or unsaturated? Yeah, you get it now, right? If you see the cane, then it's unsaturated, right? Yeah. So it doesn't really matter. For the phospholipids, it turns out maybe both are saturated, maybe it's one of each, right? And that's gonna, have different implications, but we'll not talk about it now. Okay? What's important to remember about this molecule is that the tails actually makes the molecule hydrophobic, which is what you would expect, right? Fatty acid tails are part of the lipids and it's hydrophobic, but there is a phosphate head group here, right? And that makes the molecule hydrophilic. So it's very interesting because part of it, it's going to bind well to water, the head, and then the other part is going to try to avoid water, okay? And you are probably familiar with a molecule like this in everyday life. Whenever you try to wash your greasy dishes, for example, the detergent that you use is a molecule that looks something like this. You try to wash the grease off the plate with just water, it's not going to work, right? Because water and grease don't mix together. But when you add the detergent has a hydrophilic part that binds to the water and a hydrophobic part that binds to the grease. And then it's able to cause the grease to come off of the plate along with the water.
Can you repeat that, please?
Sure.
The
So
last part that you said.
Yeah.
About
the detergent, right? Yeah. So when you add the detergent has a hydrophilic part
Okay.
and a hydrophobic part.
I get it.
The hydrophobic part will bind to the grease, and then the hydrophilic part will bind to the water, and so the water will carry the grease off the plate.
Okay.
Okay. So that's an example of a molecule that behaves like this. But what do we use it for in the body? Okay. I already wrote it down for you actually. It is a key component of the cell membrane. Okay. All the cells are surrounded by a cell membrane, and we are made up of trillions of cells, of course. And this is a cell right here, and you can see the cell membrane is made up of 2 layers of these things. Okay. So in addition to CH and O, there is also going to be phosphorus, which is part of the head. Okay, so that's what it looks like. You can see some of them are unsaturated, some of them are saturated. We will learn more about this later, okay, in lecture 4. Right, so don't worry about it for now. Okay, next lipid. What's next on the list? Do you guys remember? Wax, that's right. Wax is not on the quiz, I'll just tell you now. Right, so you don't have to worry about it. But just briefly, wax forms surfaces that are water repelling. Okay, so like the waxy coating of a leaf. That's why the water beads around them, right? Apples, your hair, your skin, they have a little bit of wax in them. Honeycomb is wax, right? People use beeswax to make cosmetics, right? Okay, like chapstick or something like that, right? Supposed to be healthier because it's organic. And basically what it is a long chain of fatty acids and a zigzag and connected to some alcohol. Again, nothing mysterious. Alcohol just means it has OH. Last but not least, we have the steroids. Okay, so cholesterol is an example of a steroid. I'm sure you heard people say, try to lower your cholesterol, and cholesterol is bad for you, which is true. Okay, there are bad cholesterol, and then there are also good cholesterol. So, but you cannot have like 0 cholesterol because your body actually needs cholesterol to function. Right? You need cholesterol for part of your cell membrane, and you also need cholesterol because cholesterol serve as a starting material to make steroid hormone. So you guys already told me, testosterone, estrogen, these are sex hormones, and those are examples of steroids. Okay? Now please make a note here. You can write it, I'm not gonna write it. Not all steroids are hormones. Okay? Not all steroids are hormones. And then similarly, not all hormones are steroids. Okay? We'll learn more about that later. Okay? But I just want you to remember that. If you use a little bit of those acne cream, right, for pimples, that's steroid, right? That's not hormone. Some hormones are not steroid. What do people need? If they have a certain kind of diabetes, what do they have in general? Insulin. Insulin is a hormone, but it's not a steroid. It's a protein. Okay. So some steroids are hormone. Some hormones are steroids. Okay. All right, that is it for lipids. Let's do a few concept check, and then it will be the end cutoff for that's good. Very simple, right? Help me out here, please. Good. Yeah. So like we put up the fingers to see, yeah, but that's okay. It's the first time. Very good. Okay, see. Anyone else? Yes. Yes. Anyone else? Good. Okay. Triglyceride, right? 1 glycerol and 3 fatty acid. The red box with the 3 tails, right? Excellent. This one is harder. We are looking for the statement that is false. Try again. Try again. Yes, very good. Anyone else? Try again, please. Look at the cheat sheet. Okay, look at the summary sheet. It's all there. Try again. Good. Try again, please. Good. 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 Okay, let's take it up. We are looking for the false one, okay? We're looking for the statement that is not true. So, I mean, since I know the answer, I'm going to start from the bottom, okay? Glycerol is a component of triglyceride and phospholipid. Let's see. Okay, triglyceride, we have the glycerol here, right? Phospholipid, we also have the glycerol. So that is a true statement. We are looking for the false one. That's not the answer. Cholesterol is a steroid. That's true, right? I told you cholesterol is a steroid that we use to make other steroids, right? Like estrogen and testosterone. So that's not what we're looking for. What do we use fat for? Long-term energy storage, right? So if you're fasting for a couple of days, the liver reserve is not enough. You got to start tapping into the fat. Okay, so that's the correct statement, which is not what we're looking for. Canola oil contains unsaturated fatty acid. What do you think? Yes, it is, right? Okay, because it's a vegetable plant oil, right? Okay, so that's unsaturated, and we're looking for the false one. So that's the false one. Okay, steroid does not have to have fatty acid. I mean, look at the notes here, right? Fatty acids are the blue zigzags, and this is my steroid. Do we see blue zigzag? No, we don't. Okay, the only key defining feature of a steroid are the carbon rings. Okay, you see a picture with 4 rings, you know it is a steroid. That is the cutoff for Thursday's quiz. Okay, please come here on time. Don't miss the quiz. If you miss the quiz, the makeup quiz will always be harder than the original quiz. Okay, so of course, unless you're sick, then don't come. Let me know. Email me. But otherwise, please study hard and you will be fine. It's not hard. I swear to you, it's not hard. Okay.
So what is the false answer? Can you repeat it one more time?
What is the
False answer. False answer.
Oh, the false one is the first one. Because steroid does not necessarily have to have fatty acid tail.
Thank you very much.
Yeah.
Sorry.
No worries. Okay. Any questions at all before we move on to proteins? Proteins are not on the quiz, okay? It's on the next quiz though. Okay. Quiz number 2.
This is quiz number 2.
That's right. Okay. So like, don't study this for Thursday.
Starts here.
Yeah, it does.
Okay. Confirmation.
Yeah, if you need to go for a walk, just help yourself, or you don't have to ask me. I know it's a lot, right? Like so much information. You need to get a copy. Tim Horton is right there. All right, proteins. Let's do it. Oh, sorry, please go ahead. You sure? All right. Protein. The main composition of proteins are our triplet again. It's always gonna be C, H, and O, but we also have nitrogen. Do you remember the 5 key elements that makes up 96% of us? Right. CHO, nitrogen, phosphorus. That's right. So we have CHO and nitrogen, and a trace of sulfur. Trace means a little bit, okay, a little bit of sulfur. The monomer of proteins, the building blocks for proteins, we call them amino acids. Amino acids. So basically, if you want to build a protein, you're going to take a bunch of these amino acids and you connect them together.
Sorry, I'm off topic, but just asking this now, is every single quiz that you make, is it gonna have that like same review?
Yeah, every single quiz is going to have a review video. Yeah. Every single test is going to have a review video. Okay. There are 20 amino acids. 20 amino acids. Okay. If you think back to carbohydrates, right? We only have 3 monosaccharides, right? Glucose, fructose, galactose. But for proteins, we have 20 of them. Don't worry, you don't have to memorize their name. 9 of these amino acids are essential. What does essential mean?
Good. Necessary for
Function, right? Yeah. However, your body cannot make them. Okay. So in order to get these essential amino acids, the only way is to eat them. So here are the 9 essential amino acids. Right? We have things like histidine, methionine, phenylalanine. If you're not a picky eater, you should be okay. You should be covered. Okay. Most of the food that we consume has a reasonable amount of amino acids in them. But if you are trying to avoid certain amino acids, then you have to pay attention. So things like avocado is really high in phenylalanine. If you're trying to avoid phenylalanine, then maybe don't eat avocado. Okay. Or if you are in need of a particular amino acid, maybe you want to, have more of one type of food. So why would you want to avoid some amino acid? Well, it turns out, here is an example of a genetic condition. That's something you're born with, okay? That's called PKU, which stands for phenylketonuria. You don't have to memorize the name, okay? This is just an example. It's one of the things they test for in newborn. Right? They do a little tube. It's called heel break. They basically have a small cut at the heel of the baby's foot, and then they collect the blood and they screen for this condition called PKU. In this condition, the baby does not have what is required to break down phenylalanine. Okay. So if they consume phenylalanine, one of the amino acids, it will build up in the brain and it will interfere with brain development. Okay. And it's obviously not a good thing, right? So they need to check immediately, and if the baby is PKU positive, then they cannot drink breast milk, they cannot drink regular formula, because those are going to have the amino acid phenylalanine in them. They need to be put on a special formula that does not have the phenylalanine. Okay? So when they grow up, right, it's not gonna affect their brain development anymore, but having too much of the phenylalanine is still not good for them. So they have to avoid food that's particularly high in those content. So this is something that's a person is born with, right? If you don't have it, then you don't have it. It's not like you can catch it later in life. Understand? Okay. Let's draw something. I will not ask you to draw this on any test, but drawing it is going to help you understand the structure. Okay, so please take a moment and copy along. Okay. Oh, and I will pass the attendance around as well. Okay, just check your name off today. It's at 21st. All right. So this is the general structure of amino acid. Okay, I'm gonna put a carbon here. See? And carbon is going to be connected to 4 things. Okay. Because of things you will learn later in chemistry, carbon can only bind to a maximum of 4 things. Okay. So at the top, we have hydrogen. And then we have, on the right side, we have C, a double bond connected to O, and then connected to an OA. Okay, like I said, I'm not going to ask you to draw this on a test, but it will help you understand what I'm trying to explain. That's a good question. Yes, you do. So this part here, this part is a functional group, okay? And the name is carboxyl. I didn't make these name up. I'm sorry if they are very confusing. And that makes it an acid. Carboxyl. On the other hand, we have N, H, and H. And whenever you have N connected to 2H here, this is called an amino group. Amino group. So that's why this is an amino acid. Because half of it is amino, behaves like a base actually, and then the other half is an acid. Amino acid. All the amino acids have this 4 structure associated with it. How many amino acids do we have? Remember the number? 20. Okay. All 20 of them is going to have this structure. What separates one amino acid from another is going to be the 4th connecting thing, okay? And we put R here as a general thing, and this is called the side chain. And the side chain is unique to each amino acid. Okay, I'll show you what I mean when you finish copying. Anyone else still need a moment? Yeah. All right. So here are the different amino acids. And you can see, I know it's a little bit different than the way we've drawn it. They have a negative and a positive. It's the same thing. Okay, don't worry too much about it. You see, they all have the carbon in the middle. They all have the hydrogen. They have the amino group and the carboxyl group. But what makes them unique and different from each other is the R group. Okay, the fourth thing at the bottom, right? So some of them are very simple. It's just another H. Some of them has rings in it. Okay, and depending on what this R group is, the property of the amino acid will be different. Some of them will be non-polar. Some of them will be polar. Some of them might even be positively charged or negatively charged. Okay. Now you don't have to memorize any of these, but you got to understand how they have different properties. Okay. So keep that in mind. We're going to come back to this later. So proteins, which are made of amino acids, have carbon, hydrogen, oxygen, nitrogen, and a trace of what? Sulfur. That's right. So that's the sulfur. And then there's another sulfur up there. So those are the only two amino acids that have sulfur. You don't have to memorize which one they are. I'm just telling you. Okay, I'm gonna show you how to connect amino acids together. And you probably will be able to do this. Okay. So let's take a look here. Okay. Let's draw it again. We have C, H. Okay, please draw it out. And then I will call this R1 for side chain number 1. Yeah. Oops. And then I will have another one next to it, which we will say R2. R2 means it's a different side chain, right? Draw those first. And then I want you to think about, using everything we've learned up to this point, how can we join these things together? All right, you know the name of the process, right? We're gonna join them together. Just try it out, okay? And then I will got it? Got it? It's hard to draw these things, right, without learning chemistry first. You will get it after week 4 or week 5 in camp.
How do you remember these?
I've been teaching for about 16 years. I probably dream about these things.
Okay, I just wanted to know, like, when you need to know that you need to put out one.
Oh, when you need to put double bonds, don't worry about it for now. Okay, like the only thing you have to know about double bond is that they are present in the unsaturated fat. Okay, that's good. So, hey, how do we connect them together? What process do we use?
Synthesis.
Yeah, something synthesis, right? Dehydration synthesis, okay? Because you're removing water, right? So, what am I gonna take from this thing? OH, and over here, H. And then the water comes out. You understand that? Okay, you always take OH from one, and then you take H from the other, right? And so what will it look like afterwards? Okay, so this one is a little bit hard to see. I mean, you don't have to draw it necessarily. You can watch me if you want. There we go. We're going to take that part. And then we're going to take this part. All right. And then you would have a bond that connects them together. So this is called a peptide bond.
Like if you take away the H.
Yes. Right. So we will put plus H2O. Okay, let me zoom out a little bit to show you one more time. So basically, it's the same theme that we've been talking about. Anytime you want to join things together for macromolecules, we have to remove the water. We have to remove the water. We'll take out the OH and the H, and now the C can connect with the N, and we have a peptide bond.
What's the difference between peptide bond and the regular bond?
They are just like special names. Okay?
Oh,
So this is
just-
specifically in protein. Yeah, in the
So it's like artificial border between Toronto and Mississauga.
sure, yeah, exactly.
Yeah.
Like even when we connect the glucose together, the bond has a name. Right? I just didn't tell you, right? It's called ether bond. Okay. Now this next part you can definitely just watch, okay? Don't copy, okay? Don't copy. So let's say I have this guy. Okay. And then what I'm gonna do is I'm gonna make a connection with another one. But we're not gonna stop there, right? Because that's just 2 amino acids. And then what we need to do is we actually need to do another one. Okay, don't draw this, just watch. I'm just trying to make a point here. And so this would be R3. And then what's going to happen next? Well, you guessed it. We can keep on doing this over and over again. And we can keep on connecting more and more trains and make it longer, so to speak. Okay, so this would be like R1, R2, R3, R4, R5, R6, and then it can keep on going and going, right? So collectively, the whole thing is going to be your protein. Right? Now, I know it's a little bit confusing here. I can see it. I can see it. Just give me a moment. I never know where my markers are. Give me a second. Oh, it's in my. Let me try to draw the parallel for you. Okay? All right. Monomers for carbohydrates, they are called mono what? You should know. Quiz is on Thursday. Mono.
Saccharide.
That's right. And then you have 3 of them, right? Glucose, fructose, lactose. Proteins. Just learn it now. What are the monomers for proteins? What did we draw on the board? What are these things called? Amino acid, that's right. Right, the building blocks, right? And how many amino acids do we have? 20. Okay, so they all have names, right? But you don't have to know them. Okay, so only 3 for carbohydrates, but 20 for these proteins. So far, so good? What about the polymer? Okay, if you connect a bunch of glucose together, this thing is called a polysaccharide. Good. And we have different examples, right? Cellulose, starch. Right? Yeah, exactly, right? Good. So what about the polymer for protein? The polymer for protein is called polypeptide. Okay. Polypeptide. It's like one amino acid to another amino acid, amino acid, amino acid. Right? It's a polymer because it's the same thing, repeating itself, right? These things are amino acid. Okay? Over here, these things, they are like glucose, for example. Did you see the parallel?
Yeah.
Okay. I do not add lipids to this table because lipid is not like that. Remember? Lipid is just like components, right? It's not same thing over and over and over again. So what I'm trying to show you here is this. That's one amino acid. That's another amino acid. And another one, and another one. And it forms the whole chain, right? Okay, so this whole thing is a polypeptide. Now, there's one more thing I want to show you, and then I'm going to give you a break. Again, you don't have to draw this. You just have to try to understand it, okay? Look at the structure. It's the same thing over and over again. Okay, you have an NH, and then CH, and then CO. NH, CH, CO. NH, CH, CO. And then it just keeps on repeating and repeating and repeating itself. So this part that's repeating, this part that is not changing, we call this the peptide backbone. We'll come back to this after the break, but that's the peptide backbone. What changes would be the side chain. Remember the side chain is different for each one of them, right? Okay. All right, break time. 5 minutes, okay? 1:37. Okay. The next thing we will talk about for protein is the structure of protein. Whenever you talk about the structure of a protein, you have to understand that there are 4 levels. Each level will determine a particular aspect of how the protein folds up. And, it's important you understand this. Because later on in other lectures, we'll come back to this concept. So let's start with the first one here, okay? The primary structure. And primary. I'll just read it to you first. Primary structure is the unique amino acid sequence in a polypeptide. You already know what a polypeptide is. A polypeptide is basically the entire protein. And if you read the amino acid that makes up the polypeptide, that's the primary structure of the protein. Right? Essentially, if you think about the amino acids as color beads, and you try to build a necklace, right? So you might take the purple one, and then the pink one, green one, and then another purple, 2 yellows, so on and so forth. When you build a whole chain, if you read the identity of each of the amino acid from one end to the other, that unique sequence is called a primary structure. Right? Every protein is going to have a different sequence. Right? What determines that sequence would be your DNA. So the DNA, as we'll learn in lecture 7, has the instruction to make the protein. Okay, you will learn how to do it. I'll give you a sequence of DNA. You will be able to build the protein based on the DNA. Right? So here's the thing. All the proteins, the recipe to make them are in your DNA. Meaning, if your DNA changes, if your DNA mutates, then your protein might be affected. Okay, we're going to take a look at that. I think in the next slide. Oh, not yet. Yeah. So that's the primary structure. We all understand that? It's just the sequence, right? The sequence of amino acids. Now, this next part is a little bit weird. Let me turn on the camera and see if I can show you with the thing here. Okay. So I have these little what do you call these things? Pipe cleaners, okay? Pipe cleaners. So this, imagine you have the beads on it, right? Like red, green, blue, whatever, right? So if I read out the bead colors, that would be the primary sequence, right? Okay. So now this thing is going to form different structure. For example, some part of it is going to curl up, and then it's gonna form this zigzag. Okay, and not zigzag. What do you call this? Like a spiral. Okay. Other parts of the protein, it might form these zigzag, right? Just up, down like this. Right? So we call these spirals, we call them alpha helix. I'll write down for you. And for these zigzags, we call them beta sheets. Okay? So the secondary structure has to do with whether the protein has alpha sheets or if it has if it has alpha helix or beta sheets. Some proteins will only have the alpha helix. Other proteins will only have the beta sheets, and others will have a combination of both. So the secondary structure is describing which one it has. So let me write down for you. I hope you understand that. Over here. Okay, so these green ribbons, those are the alpha helix. Okay, this is a Greek alphabet, okay, alpha. And then these arrows that goes back and forth, those are the zigzag, which is called beta sheets. Okay, beta sheets, alpha helixes. Yeah. Now, what holds the alpha helix and the beta sheets together would be the hydrogen bond. Okay, if you don't fully understand hydrogen bond, that's okay. I told you. Anytime I mention hydrogen bond, you just think of it as some kind of magnets attraction, right? So to prevent the helix from falling apart, the hydrogen bond stabilizes. And these hydrogen bonds are formed between the backbone. Remember before the break, I highlighted the yellow part for you? That's the backbone. That's the unchanging portion of the protein. Same thing for the zigzags. They are stabilized by the hydrogen bonds. So why do we care about these alpha helix and these beta sheets? I will give you an example, okay? I'm not going to test you on it, but I'll give you an example. Does anybody know mad cow disease? Mad cow disease, yeah. Some of you, only some of you. Okay. Well, take a wild guess. How do you catch mad cow disease? Yeah, by being close to cows or what?
Isn't it like you, yeah.
Eating, okay? If you consume beef that is from a cow that has mad cow, then you will catch mad cow. Okay.
Can you get it from like milk?
That's a good question. I don't think so. I think it has to be the contaminated meat, yeah. Okay. So what's mad cow disease? First of all, if you catch mad cow disease, there's no cure. And this is gonna be the brain. It's full of holes, and of course, like you would die from something like that. Okay. How's that? Symptoms? Oh, you would have neurological symptoms depending on which part of the brain is being damaged, right? Okay, so like you might have a little bit of inflammation, for example, a little bit of swelling, hydrocephalus, we call it, right? So on and so forth. Anyways, so here we go. It's not caused by bacteria, it's not caused by virus, not fungus, it's not caused by living things, okay? Mad cow disease is caused by protein. So you and I and the cows, we have this protein called PRPC. It's called the prion proteins. I'll write down for you. Prions. Okay, you have it, I have it. It's consists of these ribbons. Okay, so what kind of structure is this? What do we call these ribbons? Alpha helix, right? Alpha helix. Okay, so that's the normal one. And we don't really know what it does, but it's not causing us any problem. For mad cow disease, okay, the cow, they would have a misfolded version of this protein. Instead of having the helices, now it has the beta sheet. Okay, in its fold. And what happens is, as soon as you have a misfolded protein, this is like a bad influence. Okay, it will go find normal proteins and will cause them to be misfolded. Okay, so let's say this is the normal one, this is the bad one. It will come find it, and then it will cause it to misfold. And now I have two misfolded proteins, right? And then now they will go find other normal proteins and cause them to misfold. Right? It's like a zombie movie. Okay, you have one zombie, whole class is going to become zombie afterwards, right? So if you have one misfolded protein, all the proteins will be misfolded eventually. Right? So if you eat these contaminated beef, then the misfolded protein goes into your system and it's going to start changing your prion protein. Right? When enough of them misfold, they will start to clump together and kill the neurons in the brain. Right? And then you die. The name is actually called spongiform encephalitis because the brain has a lot of holes in it. Right? So it looks like a sponge. Okay, so that's an example where understanding the secondary structure of a protein is going to be important. It's one of the reasons, anyways. Okay. Now we're going to move on to the tertiary structure. Right, tertiary structure. Now, so we have this thing already. We have some helix here, we have some beta sheet. But for the most part, this thing is still flat. Okay, like you can put it on the screen and nothing is sticking out too much, right? The tertiary structure, the next level, is when this thing kind of falls back on itself, and maybe twists a little bit, and then it becomes like a ball, okay? Like a three-dimensional shape, right? It could be spherical sometimes. Sometimes it might be like rod shape, depending on what kind of protein you're talking about. The key thing to know is that this is a three-dimensional structure, okay? Three-dimensional structure. How do you stabilize it together? This is stabilized by the interaction of your side chains. Let me go back to an earlier slide to show you. Remember, each amino acid has a different side chain. Some are positive, some are negative. So if the positive and negative gets close enough, then they can form an interaction to glue them together. Right? Same thing. Non-polar parts might come together, polar parts might come together, and that allows you to stabilize the overall three-dimensional structure. Okay, you do not have to memorize any of these examples here. Just know that they basically form connection from different parts of the protein, and it holds the 3D structure together. Now, most proteins would stop at this level. Okay, once you form the ball, then it's able to perform its function, whatever it's supposed to do. Yeah.
So different type of.
Yeah, so this whole thing is made out of amino acid, right? So this amino acid might interact with another amino acid somewhere else, and then it creates a ball. What was I saying? Most proteins would end with the tertiary structure. It's fully functional once it forms into a ball. However, in some cases, on its own, it cannot do anything. Okay, it needs to partner up with another polypeptide, and together they will form. We call it a complex. Okay, so like we need one of the green ones and one of the pink ones, and now it's able to do its job. Okay, whatever its job happens to be. So sometimes it will have like 2 polypeptides. Sometimes you will have like 4 or 7 polypeptides, and all these different ones coming together, we call that the quaternary structure. And quaternary structure. All right, so this here is a classic example for quaternary structure. It's something called hemoglobin. Does anybody know what hemoglobin is?
It's the part of the blood.
Yeah, okay, and what does it do? What does it transport?
The oxygen.
Oxygen, that's right. So hemoglobin is in your red cell and it transports oxygen for you. Okay. And the hemoglobin protein has 4 polypeptides. Okay. 4 polypeptides. Okay. I don't know how much of that went in, but we're gonna fill this in together and give you a summary. This is from your study guide. If you have it, take it out. If you don't, that's okay. You can draw the table easily. All right. I'll put important. Let's go through them again and write down some summaries. Okay, so what's primary? Primary is the sequence of amino acids, right? And what holds them together? Peptide bond. All right, so for example, like you can have a red one. Let's make it bigger. You can have a red one followed by a yellow one, maybe another yellow one. And what determines the order? Like how do you know the next one is like green? What tells you that? I'm looking for a 3-letter answer. That's right, the DNA, right? Your DNA is going to give you the instruction and tell you which amino acid to connect together. Okay? So when you read the DNA code, you will know, okay, to make this protein, I need to start with the red one, and then I need 2 yellow ones, and a pink one, and so on and so forth. Okay? Your DNA is the recipe for making the protein. Every single protein in your body has a specific DNA recipe for it. Okay? You understand that? If you change the DNA, you change the recipe. Okay? And then we'll talk about what happened. So, what connects the red ball and the yellow ball together? The peptide bond, that's right. What's a peptide bond? That's a peptide bond, okay? Look, that's a red ball, the red amino acid, the whatever green amino acid. You remove the water and you get a peptide bond. You understand? And you just do it over and over and over again. Right? Some proteins are 5, 6 amino acid long. Others are hundreds of amino acids long. Okay, it really depends. Okay, so that's that. Secondary structure description, it would be forming alpha helices, or beta sheets, right? Beta sheets. Some proteins are just alpha, others are just beta. Some are a combination of both. Bonding characteristics. Hydrogen bonds. Of the peptide backbone. So if you have to draw it out, some part will look like this. Other part will look like zigzag. Next level, tertiary. Tertiary structure is the overall 3D shape of the protein. And it is stabilized by interactions of side chains. The R, right? The R group. And it's going to fall into like a 3D ball, if you would. And then, you still have the secondary structure inside, but it's all like looping back on itself, forming like this. fuzzy 3D shape. And like I said, most would stop here, but some would take it to another level. Quaternary structure, where multiple polypeptides come together to form a complex. All right. And what holds them together would be interaction between the polypeptides. So for example, you might have 2 red ones and then 2 purple ones. So that would be your 4 levels. Make sure you understand that and be able to describe it in your own words. Now, these 3 levels, the secondary, tertiary, and quaternary, these can be disrupted by changes in temperature, pH, or salt concentration. Okay. If you heat things up, then the protein might unravel. If you make it more acidic or more basic, then again, the protein might unravel. But when they unravel, only these 3 levels will be disturbed. The primary structure, the peptide bonds, these are pretty strong, okay? These are quite strong and won't be broken easily. Okay. If you just heat up the protein, you're not going to be able to break off individual amino acids. Okay. Any questions? All right, hang in there. Just 20 more minutes. Oh, okay.
I'll take it.
Sure. I'll post it after class anyways, right? So, good. Okay, the next thing to understand, why we spend so much time talking about the structure of the protein is because the function of the protein is directly dependent on the shape and the structure of the protein. It's often described as a key and lock relationship. If you change the shape of the key, you will no longer be able to open the lock, right? Similarly, if you change the shape of the protein, it will no longer be able to perform its function properly. Okay? The shape of the protein has a fancy name. We call it the conformation. Conformation. So how do you change the conformation of the protein? Well, actually, I already listed a few examples for you, right? You can change the conformation by altering the temperature, changing the pH, salt concentration, as well as the type of solution. And so when you heat up the protein, it unravels, it denatures. That's the biology term for unraveling a protein. Where do you find the protein in the egg? Actually, a lot of people get this wrong. Is it in the yolk or is it in the white? Yeah, so half, right? Yeah, the white, okay, the white. That's where you find the protein, okay? The yolk is mostly cholesterol and omega-3 and that kind of stuff, okay, lipids. But the protein is in the white, okay? When you cook an egg, it goes from kind of transparent to white, right? That's denaturation. Right? The protein is being denatured, and it's irreversible, right? Once you unravel it, once you denature it, there is no going back. Okay, same thing with the meat, right? When it goes from raw to cooked, it changed from red to brownish. The color change is an indication of denaturation. Okay. Changing the pH. Sorry. Let's talk about what temperature. Heating up is not the only way to denature protein. Okay. If you bake like a mousse cake or something, to help it solidify, you have to add gelatin, right? You guys know what I'm talking about? Gelatin, yeah, like it's a type of protein. Sometimes it comes in powder form, sometimes it smells like fish, right? You might know what I'm talking about if you bake. Then the gelatin, once you put it in the fridge, it's going to denature because of the cold temperature. Okay. And again, there's no like reversing. pH as well, right? I think it was last week we talked about how our blood pH is very narrow, the range, right? If you go too low, then it's very acidic. And now we understand why it is dangerous for your blood to be too acidic or too basic. Because changing the pH will cause the proteins to denature. If your blood pH is too low, a lot of proteins in your blood will start to unfold and there's no coming back. Okay, and that's why it's very dangerous. All right, so these are different things that would affect the shape, the conformation of the protein. I want to give you another example. Okay? This is called sickle cell anemia. Anybody heard of that? Yeah, okay, good. Anemia is a group of conditions associated with not being able to deliver oxygen in the body. Okay, so the person would suffer from shortness of breath, they would feel tired, right? And there are different ways to become anemic. Right? If you don't have enough iron in your diet, you might be anemic. If you are having blood loss, that could lead to anemic. But this one is called sickle cell anemia. Okay. I told you a few slides earlier, we have a protein in our blood that transports oxygen for us. It's called hemoglobin, remember? Okay. The hemoglobin has 4 chains on it. One of them is made up of 146 amino acids. Okay? So this is the normal hemoglobin. Okay? The sequence is like here. Valine, histidine, blah, 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 blah. Number 6 is this thing called GLU, glutamic acid. However, for people with sickle cell anemia, which is something that you are born with because of a mutation in the DNA. If you mutate, if you change the DNA, the recipe to make the protein is now wrong, right? So at position number 6, instead of putting GLU, it's wrong, and it's replaced with VAL. Let's take a look at what that means. Okay. You don't have to go back, you can just watch. Okay. GLU is right here, glutamic acid. Okay. It is a negatively charged amino acid. That's the normal one that you are supposed to have at position number 6. But because of the error, because of the mistake, it's replaced with valine, which is a non-polar amino acid. Right? So that actually changed the protein completely. And think about baking again, right? If you substitute sugar with salt, then the thing is going to come out completely tasting wrong, right? Okay, because the ingredient is so different in property. Make sense? If you substitute, white sugar with brown sugar, then maybe it's okay, right? It's still going to turn out all right. So if you substitute like a nonpolar with another nonpolar amino acid, it might be okay. But because the substitution is so drastically different, it's gonna cause the hemoglobin to misfold. Okay, so just by changing one of the amino acids here, out of 146, the hemoglobin will misfold. And that in turn will cause your red cells to become moon shaped, okay, or sickle shaped. These red cells cannot transport oxygen. Right? They get stuck in your capillaries, and it's very, very painful. And the person is going to be anemic. You understand that? So how do we fix something like this? Well, you can't really fix it because it's the genetic problem. Okay. The roots of the problem is you have the wrong recipe, the DNA code, right? So it's not treatable until up to recently, where they are using gene therapy. And that's something we'll learn in lecture 9. Okay, that's a lot of stuff about proteins. Let's take a look at what it does. We find proteins in various structures in the body. Right? Muscle is made up of protein. Your nails, we don't have horns, but like animals who have horns, the horns are made up of protein. It's the same kind of protein that makes up your nails, okay? Hair, spider webs, they are all protein. Hormones, right? I told you earlier, some hormones are steroids, some hormones are protein. The example of that would be insulin, which helps regulate blood sugar. We also can burn protein as energy source. We prefer carbohydrates, but when we need to, we can start breaking down protein as a way to obtain energy as well. And then there's something called enzymes. Okay? Enzymes. Just a quick side note here. Some people like to get the horns of the rhino, and that's why they're being hunted and being sold on black markets. Because some people believe the rhino horn has aphrodisiac properties. Okay, if you don't know what that word means, it means like it increases your sex drive. Okay, but based on what I taught you, what's the rhino horn made up of? It's the same stuff that's made up your what?
Nails.
Nails. That's right. Okay. Does biting your nails increases the sex drive? Like, I mean, if it does, don't tell me, okay? But it's the same thing, right? You know what I mean? Like it's just proteins. It's nothing special about it. There is nothing medicinal about it, right? And if people just understand that maybe the demand for it would be not as great, right? And then people won't have to hunt these things to extinction. Right. Same thing with the scale of this thing called the pangolin. It's the thing that rolls up into like a ball, right? Like an armadillo. People believe the scale has medicinal value, right? In Chinese medicine, they believe it is good for asthma, for example. But again, it's the same stuff that makes up the nails, right? It's nothing special to it, and therefore, it's just placebo effect, if anything, right? Okay. I just have 3 more slides. I know it's a lot, but bear with me, please. So on the last kind of function of the protein, right? The slide, it says enzyme. Right? So what are enzymes? The key thing to know about enzymes is that number one, they are made up of proteins. And these are proteins that will help you carry out chemical reactions in the body. No reactions can take place in the body without the help of an enzyme. Okay? And here is why. When we do a chemical reaction, you actually have to pay a little bit of energy before it can happen. That energy you pay is called EA, activation energy. I'll write down for you. Okay, it is the energy that you pay for a reaction to happen. It's like paying tuition to be in school. And often that activation energy is very high. You need to pay a lot before the reaction can take place. Okay? Your body actually cannot sustain that kind of energy demand. And that's where the enzyme comes in. Okay? The enzyme is going to lower the energy requirement for the reaction. So, think about instead of paying $2,000 for tuition, you have a scholarship and you only have to pay $700. You still have to pay, but just not as much, right? And that's what the enzyme does. The enzyme is the scholarship. Okay, it lowers the amount of energy that you have to pay. Right, so all these reactions that are happening in the body can take place at a much lower energy cost. Okay, without the enzymes, you are not going to be able to do any of these reactions. So it's a protein that speeds up, that catalyzes chemical reaction by lowering the energy requirement. So without the enzyme, that's how much you have to pay. With the enzyme, it's much lower. So let's take a look at this picture here. We have an enzyme over here, and we have some substrates. So for example, let's just call this glucose, and then we will call this fructose. All right. So in the enzyme, there is an active site. Okay. An active site is where the ingredients will fit in. So the ingredients are the starting product, starting material for the reaction. We have glucose and we have fructose, and we're going to put it right into the active site. The shape, of course, has to match. Okay, if it's a different shape, it's not going to fit. It's like that toy babies have, right? Like this one, with the star and the moon and the circle, and you try to fit the shape in. If it's a wrong shape, it's not going to go through, right? So if the substrate has a different shape, it's not going to be able to fit into the active site. Which is why, back to my original point, the shape of the protein is related to the function. If you change the shape of the active site, then the ingredients will no longer fit, and it cannot carry out the reaction for you anymore. Okay? So they will go into the active site. The substrate is just another word for ingredient. And then you will get the product. Okay. Now, let's take a look at another example to help you understand more. Let's just say this one is lactose, okay? This one is lactose. So the lactose will go into the enzyme, and the enzyme will break the lactose down. When you break the lactose down, you get glucose and what? Close. Galactose, that's right. There you go, right? So this reaction requires an enzyme to do it. If you do not have the enzyme, because your DNA is mutated and you cannot break down lactose, then the person has lactose intolerant. Right? Go back to the slide, right, where we talk about lactose intolerant. They are missing the enzyme. Okay? So all the reactions in your body, and there are hundreds and hundreds of them, each one require an enzyme. If you're missing an enzyme for one of these things, it's probably going to cause some kind of diseases. Last slide. Where do you get your proteins? Basically, if you eat meats, you're going to have proteins. Red meat, dark meat, white meat doesn't matter. If you're vegetarian or vegan, then your best source of protein would be the beans, soy product like tofu, right? Soy milk, and the nuts. They're all rich in protein. And that's it, guys. Please study hard. and I will see you all on Thursday.